This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 62, recorded on August 15th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent. I must compliment you and Elio and Michelle on a fantastic podcast while I was off at a meeting in Washington on July 31st. I found it quite fascinating.
We had a good time. We missed you, of course, but a couple of good papers, and you can never go wrong when you have good science, right?
No.
Anyway, we missed you. Good
well,
to have
thank
you
you.
back. You were traveling, right?
Yeah, I had to go up and do my other job. I administer a number of programs for the Departments of Homeland Security and Energy, and they had an all-hands meeting up in Washington. So we had 2 days of science and discussions, and anytime you interact with graduate students and your colleagues from the national laboratories, you have a great time. You feel like your head's going to explode by the time you get on the plane to come home, but it's a good feeling.
Well, today we're doing something a bit different. I am not in my usual studio. I'm away. So I thought we would try a Google Hangout to record this episode of TWiM. And unfortunately, Michael's video camera died, so we cannot see Michael. But you get the sound, and in the end, this will be released as the usual podcast. So hopefully this will all work out. Technology, Michael.
My poor eyesight camera is from, 2005, 2004, whenever they released eyesights. And so it was working this morning when I did the test, but then it decided to stop just before we started. So you can never win.
I was looking forward to watching you there, but another time.
Then everybody can see how the sausage is made.
That's what Ray says too. Did you get it from him or did he get it from you?
He probably I'm older, so he probably got it from me.
That's what he said to me yesterday when we were working on this. He said, "Let everybody see how the sausage is made," and then they won't want it anymore. Isn't that right?
That's right. That's right.
All right. We have 2 papers today and a bunch of email. Michael has one that he has chosen. It's an MBio paper. It's called Interkingdom Signaling Induces Streptococcus Pneumoniae Biofilm Dispersion and Transition from Asymptomatic Colonization to Disease. It's quite a mouthful there, Michael.
It really is, and I must admit, I first encountered this paper while listening to a story by Deborah Franklin that was on NPR last week on, I think, August 8th. And I think her title of the story was an alliteration on the popular TV show that had its summer premiere last week called Breaking Bad. And the title of her story is, "What Makes Good Bacteria Go Bad? ? It's not them, it's you." And that's effectively the essence of this paper that we're going to describe. And I think when you begin to look at seminal papers in the field and looking at how investigators tease apart a story, this paper at first looks really intimidating when you look at its really well done figures. But when you get into the paper, the figures are so elegantly simple in terms of how they ask and answer a question. And the investigators, Marks, Davison, Knight, and out of the laboratory of Anders Håkansson from SUNY Buffalo, really help teach us how to begin to tease apart this important problem. And the title that Deborah Franklin used is really for those of us who struggle for oral qualifying exam questions for graduate students, this is a great one. What makes good bacteria go bad?
I like that too.
Because it's short and open-ended, and it really can help, see how much the graduate students are reading in the field and really begin to get them to focus on what's going on. So titles are important in crafting stories to get people to read papers, but the opening paragraphs of her piece from NPR that's available on the web, I'm just going to read to the audience, and this is actually what made me go and hunt up this paper. So here's what Deborah wrote. Imagine a friend of a friend brings his family to stay with you, his family of tiny survivalists. For weeks or months, you all live quietly side by side with no problems. You share meals, your kids play together. Then one day you get sick, maybe felled by a bad cold or the flu. Suddenly, certain the end is near, your jittery house guest breaks out an armory's worth of chemical weapons. He abandons his community to save himself and hunt for a new home, wreaking havoc on the way out the door. And the data that are in this manuscript literally are those two paragraphs. And so the real question is, as you begin to approach these things, and this is one of the great things about MBio, is they have, in addition to an abstract that describes the science, this journal has something called the importance box, which helps me find papers that my normal table of contents scanning might not always locate, which is why it's important sometimes thumbing through the journals, even if they're all electronic. MBio is an all-electronic journal, and so what you have to do is you have to thumb through the all-important or the importance box to see whether or not the paper will pique your interest. So one of the things I always try to do when I'm approaching these papers is I look for the bottom line up front. And since I spend some time with the military folks, they call this bluff, bottom line up front. Uh-huh. So the first thing is their report, or this paper, addresses the mechanisms involved in the transition from an asymptomatic colonization process to disease. And if you think about it, this is one of the holy grails of pathogenesis. I think Alio had on his Small Things Considered blog once that the majority of infections actually are transition from biofilm to things gone bad, to the disease state. And so I think, and this is what really intrigued me about this paper, is that they began to help me understand how to ask this question. And so the second thing that this manuscript will do is they able to measure changes in the nasopharyngeal environment, first in vitro and then in vivo, that resulted in the release of the, quote, normal flora. And in this particular case, it's something that, if you ask the introductory medical student, is strep pneumo a normal flora microbe? Most of them will tell you no, they say it causes pneumonia. But what you learn in their introduction is that strep pneumo we're colonized with it within a few weeks of life, and it remains with us. It colonizes our upper respiratory tract, and it exists as a biofilm without causing us any problems.
But not everyone, right? Not everyone is colonized.
Oh, no, quite a few people. It's a frequent. The way they describe it in the manuscript, they use the phrase "a frequent finding in healthy individuals."
So they say 10 to 40% of healthy individuals are colonized.
Mm-hmm.
So do we understand why the others are not colonized?
No, and they didn't tap dance around. That. My guess is because of space considerations, but it made me want to go and see. Now that I know how to think about these experiments and the design of these experiments, it makes me want to go and look to see what's going on. So then, the way their story evolves, they measure changes in the nasopharyngeal environment that results in the release of bacteria that form that colonizing biofilm. And the first thing they do is they develop a model where they have the ability to do this in vitro. So you can do those very straightforward binary experiments where you have plus and minus, and you can do all sorts of neat things. And they measure gene expression, and they look for a virulence phenotype. And the virulence phenotype is very different from the biofilm phenotype. And they also comment that when microbes are grown in nutrient-rich broth, they form this planktonic form of life, which is very different. And in fact, that's their 3 experimental stages of the organism that they use. They use strep pneumo that's grown in a biofilm. They grow strep pneumo in a planktonic state in a defined medium from the 1980s called CDM broth. And CDM broth, it's in an infection and immunity paper from 1980, and you look at it and it looks like the back of a Special K box. They list all the vitamins and minerals and everything under the sun, and it's curious in that it's got 10,000 the concentration of 10,000 milligrams of glucose per liter of this medium. So it's extremely sweet, in which they grow this strep pneumo, and then they have the form of strep pneumo that has been released from the biofilm. And the way they do that is they bring one of your friends into play, influenza A, that actually causes this phase transition from going as a biofilm to where it exits. And they have very different phenotypes.
So that's biofilm is grown on human epithelial cells in culture, correct?
Correct. And I'm going to get to that because that's really the premise for this paper, is the transition from an avirulent biofilm to the diplococci form that then results in the invasive disease that is the triggering event. And what they report is that these triggering events from a biofilm form of the disease is often preceded or coincident with viral infection. And this, when I was reading the introduction, this immediately made me think of the 1918 pandemic flu that killed so many people on the planet. And, you know, there's been this great debate whether it was the viremia from influenza A or whether or not it was pneumococcal pneumonia that was responsible for the large number of deaths. And now this paper, if we can go back into 1918 and assess what fraction of that population was colonized with pneumococcal pneumonia, maybe that particular strain of H1N1, which was the pandemic version, actually caused more of that biofilm to de-evolve and then, you know, move into the system where I think a lot of the folks in 1918 died from meningitis. I think that's where they was one of the things, in addition to the frank pneumonia. But they died very quickly, which is more suggestive of meningitis. So we have this interplay between viruses and bacteria, which is truly this synergism. So the intent of the first part of the paper is to investigate the signals or triggers that promote biofilm dispersal and egress. And the way I liken that is so you're one of these individuals that has tamed a tiger, and the tiger's living in your house, and it's quite happy until such time as you stick the tiger in the eye with a sharp stick. And then that tiger goes off and decides to eat you. And so it's that egress of the strep pneumo from the biofilm that turns that what was normally a house cat just lying around on your epithelial cells into this tiger that spreads throughout your system, literally devouring everything from your lungs to your meninges. And
Michael,
so
how do they measure biofilm formation? What's the assay?
They looked at it.
Or do they measure dispersal of the bacteria?
They measure dispersal. I'm going to get to that right this second. So I'm going to describe how they developed their model. And this is the clever part of their paper. Normally, if you take strep pneumo and you inoculate it onto human respiratory epithelial cells, which they abbreviate HRECs, that normally kills human epithelial cells instantly. And so the trick that they came up with is they fixed the HRECs after they grew them in paraformaldehyde. And 4% paraformaldehyde at 34 degrees C. And so that, as anyone who's ever played with paraformaldehyde, they know that that kills tissue culture cells quite well. And then they took 48-hour-old strep pneumo that was grown in this defined CDM medium, and then they transferred to the HREC cells, and what they measured was biomass on the biofilm, and they measured it by viability. They measured colony forming units per biofilm. And so what they did is they showed that for the first 48 hours, they were growing them on these dead HREC cells, and this is in their first figure. And they show that they're growing it on the fixed cells, and they're watching they're measuring viable cells, and they use two forms of strep pneumo. One is termed D39, and one's EF3030. And they grow them on the HREC cells, and then they slowly add live HREC cells. And then the question that they ask is, does this biofilm that was established on the dead cells still retain the classic phenotype of a biofilm on the live cells? So they're asking the question, can it transition to growing on live cells, or does it immediately become this tiger? And the way they measured that is they look for the biofilm. The biofilms have this ability to resist antimicrobials, and the way they measured this is with resistance to gentamicin. And so what you see is that the microbes growing on the dead cells are resistant to gentamicin, and then and this is in panel B, and then after 48 hours of growing on the dead cells, as they bleed in the live cells, they show that it's again resistant to the gentamicin after about 48 hours. So as the biofilm begins to establish itself on the HREC cells after they have established themselves, voila, they have a model in which they're able now to grow strep pneumo in vitro on HREC cells that previously was really challenging to do, and they began to understand what was going on. So now, that's experiment one. They established their model. They're able to assess that the biofilm still has a biofilm phenotype, and so now they want to learn what signals promote dispersal, and they're still doing this in vitro. So they have a biofilm model that mimics the hypothetical in vivo phenotype. And so in the literature, they know that viruses can trigger the transition first of egress and then dispersal, where the microbe effectively goes from living, it's highly adherent to these cells, to floating about so it can wander the body and wreak havoc. And the way they do that is they simply add your old friend, influenza A. And what they measure is they simply ask the question about cytopathic effect. And they know that when a virus infects HREC cells and causes classic CPE, they begin to release ATP, and they can actually measure ATP in the medium. And that's what they actually show in one of their next panels is, they have the beautiful controls. They have the HREC cells, and then they have a model where they have the murine nasopharynx, and they measure the concentration of ATP in nanomoles. And what they see is that when you add it to their magic in vitro model, they say, does it look similar to what we see in the murine nasopharynx? And if you compare the figures, they look equivalent. PBS in HREC cells, you have about 10 nanomoles of ATP. in both the HREC, which is in the in vitro, and about 10 nanomolar in the equivalent PBS infected murine nasopharynx. And then when you ask the question, you add influenza A, which I'm just going to call flu, you then see a mean value of about, it looks about 30 units, 30 nanomoles of ATP that they're measuring, and that's equivalent across the board. So first, they're showing that their model is behaving similarly with viruses, releasing this product that's associated with a viral infection. And so now they take their model and they ask the question, does it now release the pneumococci that were attached and stuck on this biofilm? And so they have, again, two cell types of strep pneumo that they're using, EF3030 and the D39. And the way they measure this is with a simple assay of measuring for the bacteria that are dispersed into the medium. And again, they use plus and minus influenza. And what you find in the control without the influenza, most of the strep pneumo is attached to the biofilm. And when you add the influenza and wait 48 hours, it goes boom, and now you have a 10-fold higher concentration of strep pneumo in the medium or in the supernatant of their in vitro system. And so this positive value that they're seeing, the ratio of dispersed bacteria over those that are attached to the biofilm, it's again showing that their in vitro model is behaving to what is seen in the in vivo model in an animal. They already know that strep pneumo disperses when you give it a viral infection. So now they're going to begin to try to tease apart as to what that signal is. And so, the literature has, and you're probably better able to address this than I am, being the virologist, of the stuff that happens after viremia. When you get the CPE, we know that ATP is released. We know norepinephrine goes up. We know the glucose concentration goes up. And then most importantly, and I even know this from my days TA in virology, is cytopathic effect means the cell lyses and you spill the gut. You spill the contents of the cells into the environment or extracellular milieu surrounding it. And of course, that's just chock-a-block full of good stuff. So they do the next experiment where they have a control, which means without virus, and then they add these various concentrations, and they just measure the optical density in microtiter plates. So OD, something really simple that you can do, and you can do this in a microtiter plate. Put it in your microtiter reader, and you can measure the OD. And so they added these agents to a 48-hour biofilm-based culture that had been infected with H flu. And what they see, they use norepinephrine, they use ATP, they use glucose, they use the HREC lysate. They use temperature, because what's the other thing we know about viruses is they cause a fever.
Yeah.
And then they use 38.5 degrees, which is the fever associated with a virus, and the HREC lysate. And what they find is that the optical density is the highest when you're spilling the guts, which means the virus is causing the cell to die and explode. And then that's causing all these bacteria that are attached to the biofilm to wander. I mean, it's just incredibly elegant that, these signals that are coming from us as a consequence of a virus infecting us, causing the cytopathic effect, and then these bacteria begin to float in our system and they can become locally invasive. I mean, it's just incredible.
So this is an important experiment because, in fact, there's no virus here. This is just the biofilm, and they're adding
these parts. There's norepinephrine, 10
The
the
thing
10
is that,
nanomolar of ATP, glucose.
yes. So I was wondering before, if influenza virus trashes the cells, sure, the biofilm's going to disperse because they're somewhat depending on the cells to make their biofilm. They're stuck to them. But this shows that the simple trashing is not it. There's something released from the cells that actually is dispersing the biofilm. Something
Hence the title of the paper,
yeah.
the interkingdom signaling.
Kingdoms being
Humans
eukaryotes. Humans not a kingdom, but eukaryotes are. And,
Yeah.
and the other one, bacteria, right?
Yeah.
We don't use the P word anymore.
No, we don't use prokaryote anymore. We use, so, it was just absolutely elegant and straightforward. Now, this is all in vitro. So now, this is just the first 2 figures. There's 7 figures in this paper. And, when you first begin to look at it, you get pretty intimidated just looking at the figure, but then they begin to tease apart looking at other things. So they ask about gene expression because, everybody who's got an array out there has been doing gene expression, and they're asking the question about convention. This is where they bring in the 3 flavors of bacteria. That's how I'm going to describe them. 3 flavors. You got the biofilm bacteria. They're all the same genus and species. Strep pneumo, 2 strain types that they use, the 3030 and then the 39. And then they have the dispersal one, because they now know how to disperse bacteria from their model. So they can actually pull the bacteria off of the supernatant from figure 2 and add it to the system. So they have bacteria in different phases. That's really the hard thing to do, is how do you pick the population that has the right genes turned on? That's why this is such a neat paper, because their model system enables them to set the gene expression. And then their control, of course, is you grow up bacteria in a bucket of whatever, and they're using this defined medium, because that can, what you grow bacteria in can often change and skew your results. And so they then, and this is in their next figure, is they begin to look at these 3 flavors of bacteria: biofilm, dispersed, and planktonic, and they ask about these virulence factors. And they look at capsule, and every medical student knows the principal virulence factor for strep pneumo is capsule. We beat that into them ad nauseum. And of course, that's one of the first things any introductory micro course teaches when they introduce DNA, because it's the famous transformation experiment, the rough pneumococci and the smooth pneumococci. Yep. And so you begin to look at it, and that's what they take you through in figure 3 is they show these genes, the capsule cluster. They show the adhesin, and anytime you can stick, and then they show this thing called that promotes an opaque phenotype to a transparent phase. The opaque, obviously, is the transparent phase is they produce low amounts of capsule, and the opaque phase is when they're producing high amounts of capsule. And you can look at this color figure in their paper, and you can immediately see what's important for virulence and what's not. And the final panel in this figure, without spending too much time on the 3rd figure, is panel F, which looks at our factors, principally tumor necrosis factor alpha, interferon gamma, all of these things that are hallmark signals of your friends, the viruses. They look at TNF alpha, interferon gamma, IL-4, IL-8, IL-6, IL-12. I mean, it's like a lecture in immunology.
So these things are all going up in these cultures, but it looks like interferon gamma is the biggest induced there. Is that an important observation, or is just the overall pattern?
It reached significance, and they comment the populations further differed in their ability to hear, invade, and kill HREC cells, and that's effectively what they're looking at. And the dispersed ones, The dispersed ones, of course, are the evil ones, and planktonic ones, of course, always kill HREC cells. It's sort of your hypervirulent control, because what they learned how to do is effectively transition a strep pneumo so that it would then grow as the house cat in a biofilm state, and they figured out the trigger to make it go from the house cat to the tiger. And so what figure 3 effectively does is confirms that the expression data indicating that a dispersed bacterial population is indeed distinct, not only from the biofilm, but also from conventionally grown or broth-grown bacteria, showing less adherence, more invasion, because that's a hallmark of virulence, is invasion, and higher toxicity to HRECs. So this is still all in vitro. So they're laying out this story. They have their signals. We've established the signals. We now have established that their in vitro model behaves similarly from a perspective of the HREC cells responding with the appropriate cytokines. And so now they cross the Rubicon and they go in vivo.
I get it. Let me just clarify the interferon gamma. So
Okay.
they're measuring the production of these cytokines in response to adding the different kinds of bacteria to the cells, the biofilm, the dispersed, and the planktonic.
Correct.
And TNF alpha and interferon gamma are all higher than the others. But interestingly, the dispersed bacteria induce the most of those two cytokines.
Correct.
And that's the idea here, is that the dispersed bacteria, which are dispersed by flu, are pathogenic in part because of these kinds of cytokine inductions. Because these are bad, these cytokines. They help you, but they can also do bad things.
Oh, yeah. And in fact, that's one of the hallmarks associated with pandemic influenza is, they're very much worried about the cytokine storm that is induced from the viremia.
Right. Okay.
So now we now cross the Rubicon, and we're going in vivo. So again, their in vivo system is very similar, except now they're moving into mice, and so they inoculate one strain, EF3030, into the nose of the mouse, and they ask, does it colonize? And of course, as they take you through their figures, it does indeed colonize. So they establish the pneumococci in the mice, and they asked the question, does it penetrate? So what they did is they inoculated 40 PFU of influenza A in a mouse, and they had a mark level of viral pneumonia on post-inoculating at 24 hours. And when they looked, they found that the pneumococci had spread or disseminated to both the lungs and middle ears, because one of the things I forgot to tell you in the introduction is that in addition to causing pneumonia, strep pneumo is responsible for otitis media. And so they're effectively hunting to see if these bacteria go. And so then they try their magic factors. So we got no viruses now.
There's no viruses in this experiment. There's just pneumococci, right?
Pneumococci and magic factors.
And the three different kinds, the biofilm, the planktonic,
correct,
and the dispersed. And they're seeing what these do in mice. Do they colonize and do they go to other places, right?
And so then again, they come in with their norepinephrine, their ATP, their glucose, their HREC supernatant. Am I right? Is it? I know it's glucose. Oh no, it's hyperthermia. It's a hyperthermia model, and my eyes are going, and the figures are small. It's hard to keep all of these straight.
Well, figure 4 is just looking where these strains go.
They're looking where the strains go.
I think the takeaway there is that the dispersed bacteria tend to disperse. They go into the lungs, the bloodstream, and the biofilm bacteria stay put. That's 4. So 5 is some histopathology.
5 is some histopathology, and then figure 6.
6 is what you're talking about.
Yeah, figure 6 is where I was getting into they're effectively lavaging the mice, and then figure 7 is where figure 7 is where they're doing the febrile range hyperthermia.
So figure 6, before you go on, figure 6, they have mice that are stably colonized with the pneumococci. Then they infect them with influenza.
With the influenza.
Right, and then they measure the biofilm and the dispersed bacteria at different body sites, nasopharynx, lavage, lungs, and ears.
Correct.
And it looks to me, just by looking at colors, good thing I'm not colorblind because they're green and red.
Oh, yeah, it drove me crazy. I had to blow them up to very large.
Are you colorblind?
I'm red-green colorblind, so when it's really small,
Ah.
I have a rough time. But I can tell them apart if they're big. If
So
there's
one of them that's striking here is lungs on day 5.
Oh yeah. Well, day 5 is the magic. Day 5 is the magic window because day 1 post-infection, as you know, you're the virologist, you don't have that much CPE.
Right.
So you don't have all these magic factors being released. The signaling. So it's the signaling that actually, because it's a volume effect, you have to have the bacteria effectively say, okay, it's time to decamp and move. Going back to Deborah's title is, they have all these chemical weapons and they're effectively moving to another location. Yeah. And that's what's shown at day 5.
Yeah, there's a ton of bacteria in the lungs of the dispersed form, but very few CFU of the biofilm form, as you might expect.
Right. And then they go back and they recapitulated in figure 7 that it's the magic factors that are responsible. And they can actually put in some fever and do it. And, if you look at, again, they're using the same assay. It's a viable count. They're measuring the log to the base 10 of colony-forming units per tissue. So it's a really simple experiment in terms of, they add these magic factors, ATP.
They add them to the mice that have been colonized, right?
Correct.
Got it.
And so there's no virus involved.
Right. They're trying to mimic what the virus has done by killing the cells. What's being released here, right?
Yeah. So the take-home message
which is the most egregious factor?
It looks to me like it's ATP.
Huh. How about that, huh?
It looks to me like it's ATP.
NE is another one. What is
norepinephrine, which is another bad actor.
Interesting.
Oh no, it's really now. So the take-home message is that the cell-to-cell signaling inside the nasopharynx plays this key role in giving the message it's time to move, commensal survival, pathogen colonization, and host defense, if you will. And so this signaling is interkingdom. So our bacterial guests are appreciating that the eukaryotic host, at least in the nasopharynx, is no longer ideal, and thus it's time to go. So they infect your lung, you of course hack up your lung, and they spread on a fomite, and they move on out. And it's really pretty interesting. The other thing I learned is I don't know about half-lives of norepinephrine in the body, but what I learned is that norepinephrine has a half-life of 7 hours. And ATP is a danger signal for activating host inflammation and immune responses. So if I was an individual looking at things to alleviate symptoms in infections when you have influenza to prevent going down to secondary infection, I would figure out if there was something that you could add that would bind up norepinephrine or consume ATP in the epithelial layer,
right.
to control that. And I was talking about this paper with my colleague this morning, and we were talking about something completely different about C. diff rates. And you wonder, in antibiotic therapy that results in Clostridium difficile associated colitis, the epidemic that's running around the country right now, you wonder if the antibiotics that we give prophylactically for surgery or something is actually house C. diff, then recognizes it has this opportunity to decamp, because C. diff is, many of us are colonized with C. diff, and we carry it around until we take this antibiotic insult, and then boom, it becomes a bad actor where we have to resort to stool transplants often to recover. So I think this paper is significant because it establishes a model and then it recapitulates the model in vivo, showing you what they saw in vitro actually they saw in vivo. And so I was really impressed, and the figures are a little bit intimidating at first, but when you begin to take them apart, I think these figures would pass the ALEO test because they're clear, they're descriptive, and you can understand them just by reading the figure legend.
I think this two pathogen interaction is getting a lot of momentum. For many years we've been just studying one at a time in our models, but now it's becoming clear. This is one example, and there are a couple of others out there as well, that when you have two pathogens, you get different things happening than with either one alone. So here, for example, I think it would be really interesting if they infected their mice with another virus, a rhinovirus, for example. Do all viruses that cause respiratory infections release the strep pneumo biofilm, or is it just certain ones?
Yeah,
right.
And that's, is it rhinovirus? Is it adenovirus?
Metapneumovirus, sure.
Or the new one, the Saudi Arabia, the
coronavirus,
MERS coronavirus.
Oh, yeah.
And so I think they have a cookie cutter device now to ask a lot of these really neat questions, and I would actually commend this paper to your TWiM viewers.
Yeah, it is quite good for them, and hopefully they'll listen. We'll mention it tomorrow on TWiM, and maybe they can go listen to it. I agree with you. I do think this is a pretty cool. It's a TWiM paper.
Yeah,
it is.
And I only stumbled across it because as I was driving one day, I listened to the NPR story, and it had that ultra provocative title.
Right. It's interesting. NPR did it for you, huh?
Well, picking stories for this podcast is not. We have set the bar high, and
that's right.
There's a lot of papers that come out, but I'm trying to find ones that are really the gee whiz. How can I use it in explaining pathogenesis? I'm going to start teaching medical students here in a couple of weeks, and the question is whether or not they'll want to hear about this, and and how I put it on the board exam is what they're going to ask me.
Well, in general, the kinds of stories that we like to do are ones that would be picked up by a news organization, which would be a general interest story like that. So I often look in the press to see what they're talking about, and then go to the original articles.
Yeah.
For TWiM or TWiP. I think it's a good way to pick a paper. Not always, but now and then it's important, because as you know, not every science paper is reported on in the popular press.
This is true. It's a challenge, but this, as I said, came out of SUNY Buffalo. This group has been working on it. I found one typo in their paper. I do look at the references, and they had a typo in their references. They were saying to go look at 11 and 12, and it should have been 12 and 13. So they snuck another reference in there, and they didn't repaginate their EndNote library or something.
Not bad. Could be worse.
Could be worse.
Now, just coincidentally, I also have a virus story for today. And this one is an oldie but goodie. It's an old paper. Well, old, it depends on your perspective. I have two of them, 1998 and 2000. That's pretty old, isn't it, Michael?
Yeah. But
they're really good, and we can get through these in 10 or 15 minutes. So don't go away, folks. It's a concept. This is a principle I want to emphasize by telling you this story. It has to do with a bacteriophage called HK97, and HK stands for Hong Kong, because that's where this phage and all the others in the series were isolated in the sewers of Hong Kong. And this phage is very much like the more famous lambda phage. It is composed of an icosahedral head and a tail. But this phage does something that is totally unique as far as we can tell. Now, let's first just talk briefly about this capsid, the shell in which the viral genetic information, which in this case is double-stranded DNA, is held. It's called an icosahedral symmetric capsid because it is built using the principles of icosahedral symmetry. You take one or a few proteins, you repeat them many times, and you get a shell, a very stable shell. And when you repeat proteins, you get axes of symmetry, and there are 5 and 3 and 2-fold axes of symmetry on a icosahedron. And all that means is that there are 5 or 3 or 2 copies of one of the subunits around those axes. Now, an icosahedron turns out to be a very stable way to make a capsid, and it's also an economical way. You can make an icosahedron with as little as one protein repeated 60 times, all right? And you make a relatively small capsid that way. And if you want to make bigger capsids, you just increase the number of subunits. Now, the capsid has to be stable because these viruses are floating around in sewage, and all of the other viruses, there are plenty of other bacterial and eukaryotic viruses that have icosahedral capsids. The capsid gives them great stability to protect the genome as it goes from host to host. But the conundrum is they have to come apart. They have to release the DNA. If the DNA can't get out, it's the end of the game. And so viruses that infect eukaryotic cells do this in different ways. The capsid is metastable. That is, under certain conditions, it's very stable. But under other conditions, it comes apart, like low pH or in the presence of, say, proteases.
Or even copper.
Copper, too. Now, the phages have done something very different. They've added a tail onto these capsids. So the capsid itself doesn't have to come apart, but rather the DNA can go right through the tail and into the host cell. So that's a clever way of making compromise. You have a very stable capsid, and it doesn't have to come apart because it's got a tail through which to pass the DNA. Now,
and the DNA is extremely stable in these icosahedrons, or the nucleic acid, I should say, because you can have RNA in these as well. And the DNA is actually, or the nucleic acid is dewatered to an extent that it's extremely stable. And one of the ways that DNA, or nucleic acid, I should say, is disrupted is with free radicals. And when you dewater, when the nucleic acid is dewatered before it's put into these phage heads, it makes the DNA extremely stable. And the phage, because the icosahedron is such a remarkably stable entity, it's the ideal package to preserve nucleic acid for a long, long period of time. And that's why they've been able to go into, very old libraries and isolate phage off of vellum. And these books haven't been opened for, hundreds, if not thousands of years.
Right. Now, we don't know of any eukaryotic viruses that have tails, but there are some that have portals, a single opening on the icosahedral capsid, and we think the DNA goes in and out through that portal. So the herpes viruses, some of the herpes viruses, for example, have such a portal. Why eukaryotic viruses don't have tails, we don't know. Maybe we just haven't found them yet. But it may have something to do with the nature of the surrounding membranes on eukaryotes versus bacteria. Anyway, so when this phage was first studied, they found that, in fact, the capsid is made of just a single protein. It's called GP5. And in the first paper, the 1998 paper, they found that you could not disrupt this capsid by boiling in detergent, in sodium dodecyl sulfate. So if you want to see how many proteins make up a capsid, for example, a viral capsid, you add SDS, detergent, and you boil it, and you run it on a protein gel. and the proteins will separate according to their size. This capsid would not come apart under those conditions, and the proteins never entered the gel because they were so big. And so in that first paper, there are a series of very, very nice biochemical experiments where they use different proteases to digest the capsid, and the author came up with the idea that the capsid proteins are covalently linked in a very unusual way to make very, very large aggregates. Now, in a non-icosahedral capsid, being covalently linked is heresy.
Yes.
Because for a capsid to come apart, to release the DNA, the proteins can't be covalently linked. They have to be non-covalently linked. That's the whole principle of viral capsids. The proteins are non-covalently linked so they can come apart readily under the right conditions. But here is a viral capsid where it looked like the proteins were covalently linked. And in that paper, Robert Duda came up with the idea that the proteins are linked via a covalent bond between a lysine and an asparagine in the adjacent subunit. So remember, it's all one protein, GP5, and he thought that they get cross-linked, covalently linked to each other via a lysine-arginine bond. Okay, so that was totally unique at the time. The reaction, by the way, that causes this to occur is autocatalytic. The GP5 capsid protein itself is able to carry out that reaction. So the capsid protein, GP5, is the enzyme, the substrate, and the product. It's really, really a cool protein.
It's one of the hallmarks of viruses, too. HIV has one of these magic proteins, too.
Right. So in the second paper, which was published in 2000, and this has Robert Duda, in addition to a number of other collaborators, including Roger Hendricks over at the University of Pittsburgh and Jack Johnson over at Scripps. They solved the three-dimensional structure of this viral capsid by X-ray crystallography. And this shows the beautiful way that this protein GP5 makes up the virion. Indeed, they confirm that lysines and arginines are covalently linked. But what they also find is that the capsid subunit, which is GP5, it's found in the capsid in units of 5 or 6, depending on where you are in the capsid. And so if you take, for example, 5 of them, you put them together, you get a subunit of the capsid. Around the periphery of this subunit, there is a single polypeptide chain, which is not only cross-linked to its neighbor, but is also intertwined with the other neighbors. So think of a hexagon, or the hexagon, the geometric shape, made up of a single line, and think of another one next to it, only the one next to it isn't just next to it, but the lines are overlapping, or they're linked like chains. So all of these subunits, the pentamers and the hexamers, are all not only covalently linked, but they're intertwined like chain. And so that's why they call this protein chainmail.
Yeah.
It's like a fabric of catenated protein circles. Am I painting the picture, Michael?
Yeah, it's either that or it's like a screen. It's like a window screen.
Well, the screen isn't catenated, right? Take your thumb and your forefinger of both hands and touch the thumb and forefinger so that one circle is inside the other. That's a catenated.
The screen's not like that.
And you think of chainmail, the stuff.
Yeah, the chainmail would be it. Yeah, that's the best way.
So that's what this capsid is made of. It's made of protein chainmail. It's covalently linked, so it's incredibly stable. There's no way this capsid would ever come apart to release the DNA.
So it had to invent tails.
So tails, on this page for sure, there had to be a tail. And who knows if this came before or after other kinds of capsids. But if it came before, then you would say, yeah, the tail evolved to take care of getting DNA out of this kind of capsid. Because otherwise,
my suspicion is if you do the molecular taxonomy on it, you will find that the viruses had the tails before they developed the chainmail, because what it's doing is it's stabilizing the icosahedron to protect the DNA so that the phage can continue to the next generation. Right. And it's making that DNA that much more or the nucleic acid that much more resistant to anything, because the viruses, that the eukaryotic viruses are, when you compare them to the bacterial viruses, are much less recalcitrant to things. I mean, anything that can withstand boiling on SDS.
It's amazing, right?
It's protein. I mean, it's really quite remarkable.
So the question remains, and this is really not a fair question because it's a why question, but why would this virus evolve to have this kind of chainmail? And the answer is probably this capsid happens to be very thin. The shell of the capsid is quite thin. So the idea is that it would not be very stable unless it evolved this kind of chainmail. And so that may be it.
Well, the other thing is as you thin out the capsule or a capsid, you can probably pack more DNA in, or nucleic acid. And so it's a law of diminishing returns. I mean, the molecular biologists out in the audience would desperately love if AAV would take more DNA.
Right.
But it can't. I mean, AAV is stuck. And it's principally stuck because it can't stuff any more nucleic acid into its little head. And, I think that's what probably we're witnessing with this chainmail, is that they traded off the thickness of the protein in the icosahedron, and they were then enabling it to load more nucleic acid. So how big is the virus?
How big is the genome?
Yeah, is it lambda-like with over 100K? It's over 100K.
Yeah.
So it's a big virus when you compare it to something like AAV.
40,000.
Yeah.
Base pairs. Yeah.
So it's a reasonably well-sized virus.
It's about the size of adenoviruses. Yeah. Now, this was the first protein catenane discovered. There turned out to be other phages now that have similar kinds of chainmail. But as far as I can tell, I have not found any other kinds of proteins that are catenated in this way, except from phages. The first paper has a wonderful introduction. It reads, when craftsmen in Japan made katabira for ninjas and the guildsmen of Europe forged chainmail for knights, they were unaware that their armor reflected an ancient biological design used by bacterial viruses.
And definitely it came well before the Middle Ages when they were developing chainmail.
Isn't that great that this interlocking rings of metal has had an analog in phages that probably evolved billions of years before?
Well, the whole world of phage biology, I think, is going to serve as a primer for our friends in nanotech.
Yeah.
All the nano people. And, the US EPA is really struggling with nanoparticles. And I think if you look at some of the biologically based nano, you could almost make a safety argument that it hasn't killed us.
Right.
Whereas some of the other things, you don't know whether or not some of the man-made or natural, the spontaneous chemical nano things that are being made, you don't know whether or not they're going to have deleterious effects. Whereas,
of course,
these phage, we've been drinking them by the millions, literally from drinking water each day.
That's right.
And they haven't killed us yet, and they haven't caused cancer. So, maybe that is another, as the EPA begins to review all these nano things out there, they may wish to think about phage and look at some of the safety associated with phage.
Take some lessons from biology, right?
Oh, yeah. Yeah. That what does not kill us makes us much stronger.
All right, let's do some email here. Here's one from last week, Michael. We couldn't answer this, so I'm going to repeat it for you. And this was from Robert, who writes, in TWiM 60, actually it was 2 episodes ago, Michael Schmidt suggests that glucose for fermentation from biomass is a necessary step in production of fuel ethanol. Fuel ethanol and other low molecular weight compounds can also be produced by autotrophic anaerobic fermentation of syngas, which is a mixture of carbon dioxide, carbon monoxide, and hydrogen, produced by pyroclastic treatment of a wide variety of organic material, including lignocellulose, recycled tires, sewage sludge, and natural gas or methane. And these reactions have been studied for many years and are being commercialized by Cascata. Never underestimate the ability of microbes to scrounge a living from nearly any environment with water and energy available. So, have you heard of syngas, Mike?
Oh yeah, as entering graduate student at Indiana University, George Hageman was working on the metabolism of carbon monoxide, and I was of the opinion that carbon monoxide wasn't good for anything except killing people in cars. And he pointed out to me as a young graduate student the amount of energy available in carbon monoxide, because again, it's all about those 2 electron shifts. Mm-hmm. And anything that has, more reduced electric energy, you can make a living off of. And of course, the oxidation state of carbon in CO2 is its most oxidized form of carbon, so all the energy has been squeezed out of CO2, while CO, of course, still has a pair of electrons ready to go, and that's energy. And the microbial world follows the old adage, waste not, want not, and they eat everything. And so this pyroclastic treatment, that's effectively how oil was originally made, is rocks on top of all the debris after that asteroid hit the planet and killed all the dinosaurs. And consequently, it's really pretty important to look at all of these things. And, it's part of that yin and yang between greenhouse and actually fixing carbon. So if we could stop things at carbon monoxide, but the bacteria want to eat it. And
so you can make fuel ethanol with syngas then?
Oh, yeah.
Okay. We couldn't confirm or deny. So thank you. Next one is from Tim. I'm a Canadian here in the USA attending graduate school. Occasionally I hear hints in Michael Schmidt's language that he might be a fellow Canuck. Words like university and Zed, and of course his impeccable class and intelligence. Michael, thinking back, I'm not sure you've revealed anything about your past prior to your days in Indiana, at least not in the TWiMs I've listened to. I'm still working through them. If this is on purpose, I respect that decision. I don't mean to pry into personal lives. I was just curious. Happy TWiMing, Tim.
Well, I grew up in Chicago, and I've had many Canadian friends. One of my good friends is Gloria DeLille from Queen's University up in the capital, Ottawa, or on the lake there. And so I spent a lot of time with Gloria, so I think, her phraseology has rubbed off on me, and I have this inherent ability to pick up an accent, though I haven't picked up the Southern accent as much. And when I'm around my brothers in Chicago, the dems and does come out really bad. So I sound literally like a Saturday night parody when they were doing the Coach Ditka shtick on Saturday Night Live of the Bears.
Okay.
So that's no, I'm not Canadian. I just have friends that are Canadian.
But you do have impeccable class and intelligence.
Well, thank you.
Next one is from Juan, who writes, Hi, I'm currently a bioinformatics and genomics PhD student at Penn State with a master's in computer science and statistics. I'm currently working in antibiotic resistance in bacteria. I'm almost up to par with TWiM, and I'm very grateful for your insightful discussions and the time you put in teaching everyone. This has been a great foundation for my microbiological knowledge. Keep up the good work. I recently had the good luck to meet Dr. Joe Handelsman during a distinguished talk here in my department. I'm also a listener of TWiV and TWIP, but not as often as TWiM. Thank you, Juan. The next one is from Anonymous, who writes, Last year I started teaching microbiology for students interested in health-related fields. I caught up on all past TWiM episodes to help prepare me for teaching. I've learned so much from the podcast, and so much what I already knew has been reinforced. In particular, the recent mention of conjugation experiments brought back happy memories since I worked on a self-mobilizing pathogenicity plasmid for years. Listening to TWiM has an added benefit since my students think I'm much smarter than I really am.
That's always nice.
I'm from another country, and a few days ago I was talking to a colleague, also from another country, and we discussed our shock when we first came to the U.S. and found that everyone who works in healthcare can simply go home in their work clothes. In fact, they almost seem to wear their scrubs as a badge of honor. Do you see my scrubs? I do really, really important work. It is my impression that many cases of nosocomial infections are associated with healthcare staff. Consider that renovating an entire building wasn't enough to get rid of Klebsiella, which was probably brought back in on the first day of reopening by the staff. The staff carry these bacteria, fungi, and viruses in their noses, on their skin, hands, and clothes around with them. They leave work, stop to buy groceries on their way home, pick kids up from daycare, school, and take their clothes home and wash them with the rest of their household laundry. In a number of other countries, healthcare personnel have to change in designated rooms at the facility. Their uniforms have their names sewn in or embroidered on, stay behind at the end of the day, and are properly washed by the facility itself. Personnel can and sometimes are required to showers before they change into their regular clothes and leave the facility. Maybe I'm missing some crucial piece of the puzzle, but it seems like a relatively inexpensive and effective way to limit spread of antibiotic-resistant bacteria to the general community. It may also have a positive outcome on the spread within the healthcare setting. I would appreciate it if anyone on the podcast could comment on this, and there's no one better than you, Michael.
Well, he or she's got a point, and I think it's a matter that communal laundry was the first thing to go. And when I first came to MUSC, they used to wash the lab coats for the attending physicians, and that went away. And people would change out of their scrubs. I know in our surgery department, it's unwritten holy writ that the attendings change out of their scrubs and put on their, quote, street clothes. And they, of course, have access to a locker room. And I think a lot of our healthcare facilities have gotten rid of locker rooms to do exactly what the listener is suggesting. And I think it's because of absence of data. We now know that bacteria are very sticky little creatures. We know that clothing can serve as a substantial fomite. And I think it's just a matter of developing the data showing plus and minus this behavior, what it can do to healthcare-associated infections. Unfortunately, those studies are expensive to do in terms of monitoring infections. But I think, as the listener suggests, with hospital-associated infections accounting for approximately $50 billion in added costs to healthcare in old money, God knows what it's costing in present-day money, I think the Center for Medicare Services will begin to revisit many of these things. And the hospitals that are going to be paid to actually reduce readmission rates and to reduce infections are going to be looking for this low-hanging fruit. And what this listener describes is low-hanging fruit. And I think individuals in the C-suite, the people making the decisions, often never have taken a microbiology class and only anecdotally know about fomites from what they learn from their infection control professionals. And until you actually have the seminal study demonstrating that it actually works, they'll make the argument that we don't have to do laundry. So it's unfortunate, but I think the Europeans are ahead of the curve on this.
Well, I work at a medical center, as do you, Michael, and I travel often on the subway, and I never see employees in scrubs. They do walk to the local dorms, the housing, which is a block away, in scrubs, but I never see them on the subway. I never see them in the garage. So I think people, at least in my area, are pretty good about it. What do you see? You see?
We have an outstanding hospital epidemiologist here in the name of Cassie Salgado, and she strongly encourages the behaviors that the listener is describing to change out. But occasionally there are attending physicians who wear their lab coats out to the garage, and it is a substantial fomite. And I think it's coming with time. I think as we make more and more people aware, and plus I don't want to necessarily bring the multi-drug resistant creatures I work with at work and are exposed to walking the halls, I don't want to necessarily bring them home.
Right. All right, the next one is from Tim, who says, hello, Twimsters. I recently discovered the trifecta that is the TWi series, and now I can't get enough of your discussions of all things micro. For graduate students like myself, keeping up with the scientific literature can all too easily become just a means for cramming information into our brains that we feel we should know. Not only do your shows keep my mind stimulated and expose me to scientific papers that I might not otherwise read, but the informal and enthusiastic nature of the programs are just the slap in the face I need to crawl out from under the stack of papers and remind me that I actually love to learn about science and to think about the various implications that a particular finding or result might have. After listening to your conversational discussion format and your fantastic insights that tie together the biology of the organism or virus, are viruses organisms, with the environment, host, experimental techniques, etc., I'm finding new clarity in the way that I think about scientific problems and evaluate scientific papers. I can't commend you enough for what you do. Formal presentations of scientific findings are a dime a dozen. Listening to world-class scientists chat among themselves about current topics in microbiology is an invaluable resource that few people really ever get a chance to do until now. Thank you. Yeah, I think viruses are organisms because the virus is an organism with two phases, the virion, which is inanimate, and the infected cell, which is living. So I view the whole thing as an organism. There you go.
Yeah.
On a scientific note, I was recently listening to TWiM 43 and your discussion on the stable formation of caveoli in E. coli. It was mentioned that it had been previously thought that stable caveoli formation in eukaryotic cells requires cholesterol, to which Michael replied that there is no cholesterol in our friends, the bacteria.
Oh, I know where this is going.
Although this was and is true in the context of E. coli, and the paper's findings, I think, it is worth pointing out that there are several described examples of free cholesterol and cholesterol-containing lipids in bacterial outer membranes.
Oh yes, it's the exception to the rule in biology. You never say never, and you never say all.
Yeah,
and
that's right.
He's absolutely correct. That as soon as it leapt out of, one of the comments is we're spontaneous, and we're not. I wasn't certainly trained as an attorney, so I don't always parse everything I'm saying to think if it's perfect, and. We especially like to be called on the carpet, and that's half the fun of appreciating the fact. And I think that's what I like most about microbiology is that you always find the exception to the rule.
So he cites Helicobacter, Mycoplasma, Ehrlichia, Anaplasma, Brachyspira, and Borrelia. In Borrelia, cholesterol-containing compounds have been shown to form organized lipid rafts in culture and in animal-derived organisms with physical properties similar to that in eukaryotic membranes. And he discusses a recent PLOS pathogens paper on that topic. I highly recommend looking into this story maybe as a TWiM topic, as it provides a shift in the thinking about lipid rafts, expanding their biological relevance to prokaryotes and could have implications for the evolution of the eukaryotic cell membrane structure. Additionally, transfer of antigenic lipids from bacteria to host cells could play a role in pathogenesis, having multiple consequences for the host immune response and potentially contributing to heightened inflammation and perhaps even direct targeting of the cells themselves by immune effectors. I've provided the citations and PubMed link to a few papers on the Bieberg-Darfuri story below, but also recommend digging into the story for H. pylori. I've rambled enough for now, as I should run. I've got to get to iTunes to write a good review or two.
Oh, that'd be nice.
Thank you, Tim. And one more from Jenny. Jenny here, RN. I've just seen something from Medscape that I thought you'd be delighted to know. An offering of clinical guidance on a successful recipe for fecal transplantation, biotransplantation for C. diff. historical background on fecal microbiota transplantation, and a picture of success beyond C. diff to inflammatory bowel disease, irritable bowel syndrome, idiopathic constipation as well. Fecal transfer appears to work for C. diff that relapses. Aside from fairly direct fecal transfers, this synthetic poo is described. These investigators created a human synthetic stool mixture by culturing the stool microbial diversity of a healthy 41-year-old woman and brewing a mixture of 33 different intestinal bacterial isolates in pure culture. They named the synthetic stool mixture Repopulate.
Oh no.
Really?
Well, it's memorable. It's memorable, and the U.S. FDA is right now looking very carefully on how to qualify stool transplant donors.
Mm-hmm.
They're looking aggressively at how do you, especially when you don't know the donor, do you look for parasites? Do you look for viruses? All the viruses that cause hepatitis.
Right.
Look at the parasites that you'd have to concern yourself with, as well as some of the other bad actors that some of us have in our guts that are, fine for us.
Sure.
And so the FDA is taking a very careful look at doing this. The insurance companies, some pay and some don't pay. And the lab tests, of course, have not been standardized. And so this synthetic stool may be a first way of doing it, but the problem is the human stool is comprised of at least 1,000, if not greater numbers of microbes, and who's to say which one's right and which one's wrong?
Sure. No, that's a very important question, and it's not an easy one, so it's going to be a while to sort out. Jenny continues, the bacterial mixture was infused into the colon of 2 patients in their 70s, both of whom were infected with a hypervirulent strain of C. diff and who had failed at least 3 courses of antibiotic therapy. Both patients returned to normal bowel patterns in 2 or 3 days and remained symptom-free for 6 months. At that time, ribosomal RNA sequences representing the Repopulate mixture made up 25% of the gut bacterial population, and she provides a link to this story. Thank you once again for making the world safe for microbiota, Vince and friends. Yours with warmest regards, Jenny.
Well, that was really nice.
That's cool. We'll be seeing many, many more of these stories until it becomes routine, right, Michael?
I think so. And, we've seen it for C. diff. We've seen it for some of these simpler bowel complications. But, there are other things out there that are inflammatory, like Crohn's disease. And, that may be something that is thought to have a microbial trigger. And Crohn's is principally an inflammatory pathology that goes really out of control. And so trying to understand what's actually going on and whether or not a stool transplant could actually alleviate some of that chronic inflammation. And, there are a lot of bacteria that calm inflammation in addition to causing it. So I think that's one of the things to, keep looking for as we scan our titles, looking for cool stuff to talk about.
Yep. This episode of TWiM, like all the others, will be found on iTunes and also at microbeworld.org/TWiM. If you like what we do, one of the ways that you can help us is to go over to iTunes and either rate the show or give us some stars there. It helps to keep us visible in the very crowded Apple Podcast directory. And, of course, we love getting your questions and comments. Send them to twim@twiv.tv. Michael Schmidt is at the Medical University of South Carolina. Thank you for joining today, Michael.
I enjoyed it, Vincent. We missed our good friend and colleague, Elio, but I assume he's having a delightful time. He's probably having a delightful lunch as we speak in Europe. So he's cruising the rivers of Europe, I think he said.
Is that right? Well, he will be gone the rest of August, so it'll either be you and I or another person or two, if I can scare someone up, to join us for the rest of this month.
Then September, we're off to ICAC.
That's right.
We're going to do TWiM at ICAC, but we're going to call it ICAC Live.
Yeah, that's right, but it's really a TWiM. We have a couple of cool guests. We have actually a bacterial, an E. coli guy, and a rhinovirus guy lined up for that show, so that should be interesting.
Well, that's good news.
I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, in particular Chris Kandian and Ray Ortega for their technical help, and especially Ray for helping us to get this hangout going, although I don't know how well the video worked, but I could see myself, that's for sure. The music we use on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.